Hello, this is your video setup for lab number 4, our cellular respiration and metabolic rate lab. So to get us started, you need to first pay attention to the formula that we are going to use, because you are going to use potassium hydroxide pellets, this solid, and the crickets and the mealworms that we'll use as they do cell respiration will produce CO2 gas. The key thing is that gas is going to be converted into a solid, and then that is essentially going to create kind of a negative pressure that causes oxygen to be consumed through this tube. And because there are markings on this tube, we will be able to measure how much oxygen they are consuming. So that's kind of the big picture, zoomed out aspect of the lab as a whole. So if you look at the setup, you've got these 3 respirometers. The respirometers that you will start with will be empty. You have to construct them, but they look like this. It's a glass tube with a washer on the bottom to kind of weight it down. And the first thing you guys are going to do with these 3 respirometer tubes is you are going to make a potassium hydroxide cotton sandwich. So I will be coming around with the solid potassium hydroxide pellets. I will give them to you kind of by pouring them out onto your lab bench. And then what you are going to do is take a piece of cotton. 1 piece of cotton goes in the respirometer. You are going to pack it with this wooden skewer. Pack, 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 pack. And you do want to pack it relatively tightly. Don't obsess over it, but you don't want it to be totally loose. Then using these forceps, because we're not going to touch the potassium hydroxide with our hands, you are going to take really anywhere between 5 and 10 of the pellets, add them onto that first layer of cotton, and then take a second piece of cotton, tap, 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 and make your potassium hydroxide cotton sandwich. And the reason we're doing this is because you are going to put mealworms and crickets in here, well, either one or the other, not both at the same time. And they have the instinct, especially the mealworms, will want to burrow into the cotton. And if they touch the solid potassium hydroxide pellets, it kills them. So you want to pack this top layer of cotton tight enough so that if they attempt to bury themselves, they cannot do that. And then, once you have all 3 respirometers set up, and my recommendation is that you do all 3 first and kind of have all 3, completed and out on the table. And you can see here I've already gotten my other 2 done. After this point, you can go ahead and put the millimeter tube. This entire tube is 1 millimeter. You can see the markings on them. It's kind of hard to see in the video, but every single line is 0.01 milliliters. And they are, there's a larger line separating the tenths of a millimeter. So one of the respirometers will be the control. That does not have any organisms in it. So that respirometer can just be assembled as is and set aside. You don't have to shove the cork down into the glass tube because they do break, they crack. So just, you want it to have a good seal, but you don't want to like manhandle it in there. For the other 2 tubes, you are going to put mealworms in one and crickets in the other. And they will be on your lab benches in these 2 containers. And you need to find the mass of the crickets and the mealworms. Not every individual one, but the total mass of all 5 together, because you'll have 5 of each. And if you look on here, you can see I've already labeled the mass in grams of the container. So when you put this on the scale, imagine there are some mealworms in here. When you put this on the scale and it reads, 15 grams, then I would subtract 11.16 from my 15 grams, and I would record the mass of the mealworms themselves in my lab. And that way you're not trying to put individual crickets or mealworms onto the balance. That would be chaos. So this is the vial the crickets will be in, and similarly, the mass of the entire container, including the cap, is already written on there, so you will just subtract that out when you put the crickets in the container on the balance. All right, there's a spot in your data book to record that information on the left side of your data table. And then, and this is where you've just got to do it, you're gonna take one of your two containers. The mealworms are easy. You can just slide them in there and they'll just go right down. Put one of the respirometers on, set it aside. Whoops. For the crickets jump, so my recommendation is you unscrew the cap, perhaps put your fingers on it, and then invert the setup, because the tubes fit inside each other, and then you can just invert it and dump the crickets into your chamber, and then put the cork on the top. All right, so now you've got your 3 completed respirometers. I'm gonna pick you guys up, and you are going to allow them to acclimate to the temperature. So some groups are gonna have this room temperature water, other groups will do cold, and you'll have ice water. But either way, you are going to set your 3 respirometers in the water, submerged. They will be covered in water, such that the tip of the respirometer is resting on the ledge of the tray. And you're going to do this with all 3 respirometers. And at this point, your job is to just let it sit for 3 minutes to acclimate the temperature. All right, because the temperature of the air in the chamber needs to cool down to match the temperature of the water. And there is a blank sheet of paper underneath here in the hopes that it helps you kind of see the markings on the tube better. It's just to help you visually contrast the markings and the readings on the tube. I hope that's the point of the paper. After 3 minutes, so let's assume 3 minutes has passed, you are going to lower the tips into the water. And at this point, and to be thorough, I probably should have already been doing this, we are not going to touch the setup with our hands. So you can actually use the skewer to adjust these, because touching these with your hands is going to add some kinetic heat energy to it, which we don't want. We want to not add our body temperature to these tubes. This one's floating around a little bit, but once something is in there, hopefully it settles. If you're having an issue, kind of like I am, getting these things to stay in place, you can take this blunt end probe and you can gently lay it on top of the millimeter tubes, and it'll kind of hold them down for you. And we're just gonna let this sit for 3 additional minutes. But in these 3 minutes, what I recommend you do is, using your little skewer to help you, your job is going to be to find the air bubble, so that when your 3 minutes is up and you start your 20-minute timer, you will know where to start. And I am trying to find an air bubble for you that, let's see. I don't know how well this is going to work, but there is the boundary right here from this point to the tip is water, and from this point towards the rest of the chamber is oxygen or air. This bubble is slowly going to move up the tube, and that change in volume is what you are going to measure over the span of 20 minutes. All right, remember, as the organisms are doing cell respiration, they are exhaling carbon dioxide. That carbon dioxide gets captured by the potassium hydroxide pellets in the cotton. It converts it to a solid, which creates essentially a negative pressure that is going to pull, if you want to think of it like that, water into the tube and displacing the oxygen as cell respiration continues, right? So I hope this helps. Again, pay attention to the background reading, pay attention to the equation, and be ready for some live animals. See you then.